right? And we'll start with our first morning panel, which is all about organoids and in vitro generation of more organized cell types. And I think this is something that I can see is taking like a really good momentum now, and just exemplified by my lab, and I'll talk a little bit about that. We were really excited in the beginning to have neurons in a 2-dimension that can form synapses and perform lots of tricks with those neurons, even innervation of motor neurons in a muscle cells. But this was always in 2D. Then, I mean, we start seeing some of these applications in 3D, not only for the brain, but other tissues as well. People in the lab really got excited with this idea that perhaps we can model that. And the truth is that the cells can do it by themselves. I mean, sometimes, yeah, you can generate a scaffold, you can do like 3D printing, but for the most part, at the beginning, I mean, the cells do it by themselves, which is really good, really save us time and trouble. So what we decided to do is to give you like just a flavor of different applications of these organoids, and we'll kick off with my talk from people from my lab who actually are improving or generating a novel protocol to generate these mini brains, as we call them, in a dish. We'll then have Tracy, who is a Ph.D. candidate, who will talk about her work as well. And Juan Carlos Belmonte couldn't be here, so his talk would be replaced by a postdoc in his lab, who really just got like a very beautiful paper in cell stem cell, and going to be here like first time here. So I will start with the work that we do in my lab with the brain organoids. And again, the whole idea that we have in the lab is really to use the iPSC so we can complete the cycle of drug screening. By reprogramming cells from patients. Most of the patients that we work with are patients that have neurological disorders caused by a genetic, well-defined mutation. So we reprogram these cells, and then we make neurons and glia cells, or the relevant cell types that we have in the brain. And we look for phenotypes, trying to differentiate the two groups, the control group and the disease group. And we look for several phenotypes at the cellular level, number of branching synapses, as well as at the molecular level, if there is any difference in gene expression, something like that. And the beauty is that you can really increase in complexity by adding other cell types, by mixing different genotypes, or by creating these 3-dimensional structures that I'll mention to you. So if you're lucky enough to have any difference between that, you can use the system as a drug screening platform and see if you can rescue, revert, or ameliorate any of those phenotypes and perhaps speed it up drug discovery in drugs that enter clinical trials. So this is the whole concept that we work with. And what I'll tell you right now is a little bit descriptive, and then I'll show you two applications of this technology that we have. So this is a novel protocol to generate these 3D brain organoids that was developed by two postdocs in the lab. This is Kleber and Priscilla. And they really started from iPSCs that can be dissociated in single cells under specific conditions and let themselves to aggregate themselves in an already like a neuroinduction phase. And after a while, so this is like a week or so, you already start to see some kind of organization in these balls that are just floating around in these dishes. So you can see that, let me point here, small units of proliferating cells. These are Ki67 cells that will proliferate, and already some glia cells or some more differentiated cells that also come along. So this was really the early stages. We already see units of progenitor cells and more differentiated cells. Then you start really inducing maturation as well as proliferation of the cells, and those units become a little bit more clear. So in the immunos, we definitely see that those proliferating zones continues to produce dividing cells, and the cells would migrate out of these proliferating zones and start to differentiate into neurons. So these are the green new and positive cells that we have in those units. And then, I mean, we have a last piece where you can really induce a final maturation of those cells. And we are now here really pushing hard to have something that's cortical specific. So we're really interested on disorders that affect the cortex circuitry. So we are trying to avoid degeneration of other brain parts, at least for now, and focusing on the cortex. And it's beautiful to see that over time, I mean, this the size of these cells really increase in diameter as you mature at different stages. So this can go up to a year, and they still do fine. And you can see at naked eye. At these stages of late maturation, it's funny to see that these organoids actually start to fold, so you can appreciate some of these foldings on the borders of these cells. And not only that, but by doing this immunostaining, you can have beautiful both radio and tangential neurons that will guide the migration of the different subtype of neurons that will make your cortex. I'll show some of the cortical organization here. So again, we continue to have a proliferating zone where the cells are dividing, and then these neurons would migrate more to the periphery of those units, of those balls or mini brains. And we have been using lots of techniques, not only immunostainings, but also single-cell analysis, really to characterize the type of cells that are being generated from these brains, mini brains, at different stages, and try to compare what happens in vivo, in uterus, or just post-birth. And I'll skip some of the data, but just show you some nice images here. So these are, again, one of those units that are SOX2 positive cells, and you see that some of the cortical neurons migrate out and start forming layers. So this is CTP2. And those neurons start to get connected and really form a beautiful network. So again, this is another one with another cortical marker, set up B2, and this is synapsing punctua, so they start to form synapses. I'll show you some functional analysis on that as well, and another different cortical layer as well. So as these mature, I mean, we can start to appreciate a little bit of the cortical organization by doing like tangential cuts or analysis on using different markers for different subtype of cortex. And yeah, we don't really have like a very clear definition of these different cortical layers, but as they mature, this becomes more and more clear. So this is just a couple of markers of the deeper and more superficial layers as well, so we can have them all. And at the functional level, I mean, these really start to become functional at very early stages, which was also a big surprise compared to the 2D system, where we really have to wait at least 8 weeks to see some kind of activity. So in 2D, you can actually do the imaging in 3D and see that there are calcium transients coming from these neurons, suggesting that there is some kind of network. One thing that is really cool about this protocol that was also a big surprise is the fact that once these neurons mature, you can go back to 2D by dissociating these cells and replating them. And this is what we have. So we are looking for CTP2 positive cells, which is a subtype of a cortical layer, layer 5 and 6. And this is one of the important layers for autism, schizophrenia, or other disorders that affect the social brain. And this is a 8 weeks protocol, common protocol in 2D. This is the same time in 3D. So the number of mature cortical CTP2 positive cells is really increased. The fact that you don't see as many MAP2 process here is that this is a picture just after replating them. So after you dissociate, some of this process is broken up, but as you replate, they will grow that again. So they become really highly arborized. But as a quantification for the number of CTP2 positive cells, you can already appreciate that you can get more at the same time as in 2D. So that was really interesting to us, and we got really excited. And then we move on really doing an analysis, an electrical analysis to see if those cells can have some kind of network. The way we do that is by using a multi-electrode array where we can just place the cells on top of these arrays. So this is 8 by 8, 64 arrays, and you just capture the electrical activity over time. So this is what we have in 2D. The different colors here is the same iPS cells that was differentiated twice. So you can see that the 2D gives you some kind of variability. For some reason, in the second time, I mean, we couldn't get the same increase in activity as we did for the first time. So actually, this is not a surprise. Any stem cell lab will have this kind of data. That's why we repeat that many times to be confident. But it's painful because you can do that and really not get much data or have to do repetition. So it's time consuming and you spend lots of money doing that. But eventually, yeah, even in the 2D, we have a nice synchronization of these networks, which is really good to see, really helpful, so we can study whenever the system works. In 3D, we're really surprised on the consistent and reproducibility of these readouts from the multi-electrode array. So this is, again, I mean, 2 iPSCs doing in different times, and it's quite amazing that they mirror each other over time. So number of spikes, number of bursts are very similar, and then the synchronization ratio is also very similar. So we really got excited to see something like that. And really, I mean, this is the type of data that we have. This is 8 weeks, 10 weeks, and 12 weeks. What we are looking is not only for the number of spikes, which is each trace here, and bursts, which are lots of spikes in a very short period of time, but also the synchronization rate is when the network actually gets really synchronized. So this is what we have been working with and decided to apply to the study of human genetic disorders. And I'll talk about 2 different neurological diseases. So one is Aicardi-Goutieres syndrome, or AGS. So as working with some neurogenetic childhood conditions, I think this is one of the worst syndromes I ever seen. Because it actually starts before birth with a really massive neuronal loss. So those neurons, they cannot really differentiate well or survive in the brain. And as a consequence, we have lots of things that happens in your brain, such as calcification, as I mentioned, severe neuronal loss that will lead to a macrocephalic brain. And most of the motor conditions and actions would be lost. I mean, those kids, they work as a vegetal. I mean, we can actually see that they may respond when mom will say something, and you can see that the kids will try to turn their eyes or will have some water in their eyes. That's how we see that they have some connection in there. Apart from that, I mean, they don't do much, but they continue to grow and to lose neurons over time. So this terrible disorder is caused by mutations in a gene called TREX1, which is an enzyme that degrades foreign DNA. And we thought that perhaps because of the early onset of the disease, the embryonic onset, that would be like a nice case or really a low-hanging fruit to show off that we can model that using these mini brains. And most importantly, if you knock down the TREX1 gene in a mouse model, you don't have the neuronal phenotype. So this is a condition that it really manifests on a human background. There is no animal model for that. The way we did that was to reprogram cells from the patient and generating both neuroprogenitor cells, astrocytes, and neurons, and the mini brains that I'll talk about. We also create cells by using CRISPR, generating artificial TREX1 mutations in a normal control background. So we have isogenic controls. So this is just an example of, for some reason it's not here. Yeah. Anyway, so we have at least 3 mutants in 3 controls, and some of those are isogenic controls. So the data I'm showing here is just these different stages of maturation that I told you from these mini brains or organoids. This is a representative picture of 24 hours, the proliferation phase and this maturation phase. So if you don't have TREX1, if you knocked out TREX1 in these cells, you can see that there is some proliferation, but it's dramatically reduced over time. So one interesting thing is that you can actually use this system to find drugs that may prevent or rescue that. And what we have here is just one example of HIV drug. This is reverse transcriptase inhibitors that can actually rescue this neuronal loss. And you increase the diameter of these mini brains in vitro. This is another drug that didn't work. So we can actually create hundreds and hundreds of these mini brains from patients and really use drug screening and simple readouts such as diameter or size to find drugs that may be helpful in repurposed drugs that already exist for this condition. And this is all because those progenitor cells, they actually die by apoptosis. And this is a readout that we have using cleaved caspase-3. So again, this is one of these mini balls or mini brains from a control cell. This is the knockout. The dots in red are the cells that are dying by apoptosis. If you use the drug, you can rescue that. This is the drug that did not work, and we can just quantify the number of caspase-positive cells. So it's relatively easy to do that using any software, imaging software. So it's an easy drug screenable readout. So the other condition I would like to talk and to mention to you is something that I got involved with quite recently, which is the Zika virus. So this is not a genetic condition, but an environmental condition that can also lead to macrocephaly or some of the most dramatic neurological problems. So I really got involved because I'm actually from Brazil and have been following the news over there and seeing all these dramatic pictures of macrocephalic babies. I thought that perhaps we could use these models to somehow contribute to that. And we really set up a very nice platform to study the impact of Zika virus in vitro. And I'll show you what we did with these organoids. So this was the work from Isabella, a postdoc in the lab. And what she did was really to expose these organoids, the cerebral organoids, to the Zika virus and really quantify the types of cells that can actually survive and die. So this is a comparison between the Brazilian Zika virus, this is the African Zika virus, and this is a yellow fever virus who does not cause any neurological problem. And this is the quantification of the percentage of cells that survive. This is TBR1, is one of the cortical layers. So there is a dramatically reduction in the number of TBR1 positive cells after infection with the Brazilian Zika virus. We don't see the same reduction with the African Zika virus. Again, telling us that those viruses, they behave differently when exposed to a human cell. And again, as a control, nothing with the yellow fever virus. So she quantified different other cortical layers just to show that this is actually happening. And the reason why is because those viruses are actually affecting the dividing cells. So in this graphic here, you have the reduction of the number of Ki67 positive cells. These are the dividing cells in these proliferative zones. And again, upon infection with the Brazilian Zika virus, there is this significant reduction in the number of surviving cells. So that may explain how the macrocephalic brain appears in those kids. To validate that, we actually set up an in vivo model where we could see vertical transmission. So the way we did that, go back. the way we did that was to infect pregnant animals with the Brazilian Zika virus. And we did that in 2 flavors using a white type C57 black 6 animal, which has like a normal immune system, and also an animal called SJL, which is also a white type animal, but has a reduced interferon response to the virus. So in this case here, what we see is that upon infection, the virus can actually cross the placenta and really affect the fetus. So we have some macrocephalic pups that were born. And this is just the analysis of the cortical thickness that we see. Again, a dramatic reduction in some pathological information indicating that these cells, similar to what we see in vitro, are actually dying by apoptosis. We definitely see some other things that is happening in those brains, and we do believe that the consequences of the Zika virus to the brain, it's much more broader than just a macrocephalic brain. This is perhaps the most dramatic cases, but even in non-macrocephalic cases, you can see that there is a consequence, and the clinical data is just coming along to show that this is actually true. So I'll stop here and to take any questions, but this would be my take-home message. This mini cerebral cortex model that we have now can be used to generate or to understand milestones, to recapitulate the milestones of the human fetus brain, and we continues to characterize that. You can use that to model a genetic neurological disorder such as Aicardi-Goutieres on patients with TREX1 mutations, or you can use that to understand the contribution of the environment into this brain neurodevelopment. So this is what we did with the Zika virus. And these could be used as a drug screening tool, not only to find new drugs, but perhaps to repurpose drugs for immediate use in clinics. So the mini brains were actually developed by these 2 postdocs. This is Kleber, this is Priscilla. The TREX1 work was done by a grad student in the lab. This is Charlie, together with a technician who became a master's student. This is Leon, and all the Zika virus study was performed by Isabella in the lab. So I'll be happy to take any questions. Thank you.
Very nice work, Alysson. Over here. Hi. So I have 2 questions. For your Aicardi-Goutieres patients, did any of them have an ADAR mutation? You mentioned Trex, but they can have other than TREX1, they can have other mutations.
Yeah, they do. Yeah, there are some patients that do have other mutations in genes that are also involved in this interferon response. And we just haven't done that with other mutations. We have been focusing on the TREX1. I do believe that they would probably behave in a similar way.
The other question I had about your Zika virus, it's really great that you can do the mini cerebral cortex studies. Have you looked at adult neurogenesis or neuronal progenitors from adults in the IPS way that you've pioneered?
That's a great question. There are other people doing that. Yeah. We haven't focused on the adult brain. There is a recent paper from the Gleason lab showing that, yeah, there's probably a consequence, but there are certain conditions where this might happen. I do believe that if you have a susceptible population, perhaps someone that has an immunosuppressed system, they might be like more real target for the virus. But we are just discovering these now.
So, just to follow up on the immune response and the role there for the Zika, I was trying to define whether the differences in the mice was one was more immunosuppressed or more immune reactive, and what is the role there in mediating pathology? The other part is, in your organoid system, have you looked into adding immune cells, particularly microglia, into those structures?
Right, so the animals that worked the best was immunosuppressed. There are other models where you knock out interferon response, so they respond even better. The virus can basically kill the animals. In vitro, yes, we are trying to add that. We're trying to add the microglia, perhaps by infuse the microglia in these mini brains, and then create a model where we can study the response coming from these cells as well. So this is ongoing in the lab.
Thanks.
Hi, I'm just wondering, in the case of improved reproducibility with the 2D and 3D culture, have you also looked at parameters such as spiking rate and bursting and duration, for example?
Yeah, so
Does that improve too?
Yeah, they basically start earlier to show that in 3D compared to 2D. The 2D eventually will get there, but we see that at one point there is a plateau and cannot really keep those 2D cells in culture for so long. They start to detach, especially on top of the multilateral array. So there is a limit that we can keep them. But the 3D is really nice because, I mean, yeah, it's from the beginning you already have all the spikes, all the synchronization in a very short period of time.
But does like parameters such as spiking rate and bursting rate also becomes more consistent?
Yes.
Okay,
Yeah.
thanks.
So, one more question here. Senator.
I'm very curious about your organoids because they look quite homogeneous. The size is quite homogeneous.
Yeah.
I've never seen anything like this. And the second point is, I was wondering whether the cortical structures that you see in the organoids is a single cortical structure in one portion of the organoid, or whether you have multiple cortical structures.
These are great questions. There are multiple structures in one organoid, and they are very similar on size. This is one thing that surprises us as well. So what I think we probably achieved, not that we're looking for that, but the way we rotate those mini brains in a dish, there is certain rotation that will keep them, the sharing forces in a way that they all end up with the same size. Yeah. It's very different from other protocols. I mean, we try other protocols, and we get a huge variability as well. So I think that's the beauty that we are contributing now, is to add a little bit more consistent on this protocol. Yeah.